Good morning. First, I want to thank you all for being here in class today. For today's agenda, we'll have a review, and then I'm going to discuss about an overview of transcription and translation, and you will do a simulation after class so that you're going to understand the topic better. Last meeting, we differentiated DNA to RNA, and we said that DNA is double-stranded while RNA is single. DNA has deoxyribose sugar, while RNA has ribose. The bases for DNA are cytosine, guanine, adenosine, and thymine. While in RNA, it has the same, however, instead of thymine, it has uracil. Both of DNA and RNA contain sugar, phosphate, and bases. Now it's time to understand exactly how this molecule codes for a particular organism works. How is that a single cell containing a specific set of genetic material will result in the development of a fish, a cat, or a human? At the end of the discussion, you should be able to answer the following questions. How do we get from the bases found in the DNA to amino acids? How do we get from a bunch of amino acids to proteins? To understand the phenomenon, we must learn about transcription and translation. This is the collective process by which the genetic code is read by the enzymes to produce all the proteins in an organism. Genes in a DNA contain information to make proteins. The cell makes mRNA or messenger RNA copies of genes that are needed. Then the mRNA is read to the ribosomes in the rough ER, and protein is produced. The key players for these processes are the mRNA. It carries the information from a gene in the DNA. Ribosomes, made of rRNA, consist of subunits and carry out an enzyme-like role. The transfer RNA or tRNA carries specific amino acids to the ribosomes. So how does this work? The first step is called transcription. There are three steps in transcription. The first one is initiation. RNA polymerase is an enzyme responsible for making the mRNA copies of genes. The DNA unzips at the site of the gene that is needed. The second is elongation. The RNA polymerase matches bases in the sense strand with RNA bases, building a strand of mRNA that carries the information encoded in the DNA. Once the RNA polymerase reaches the end of the gene, termination occurs. Encoded in the DNA is a signal telling RNA polymerase where to stop. Transcription ends at that point. After a few quick modifications during RNA processing, which is the conclusion of transcription, it will leave the nucleus, where all the genetic material or chromatin is, and move into the cytoplasm, where it will find a ribosome. This is where translation occurs. Now the second step is translation. Translation will occur inside a ribosome. The small ribosomal subunit binds to an mRNA, this one, and an initiator tRNA which adheres to start codon. Then the large ribosomal subunit joins to complete the translation initiation complex. Then the tRNA that corresponds to the next codon after this start codon will enter the ribosome. This will carry out with an amino acid which becomes covalently bound to methionine from the initiator tRNA. The ribosomal unit binds to mRNA, where the code for MET is located AUG. The anticodon is UAC of the tRNA matches the start codon and mRNA AUG. Again, the ribosomal unit binds to mRNA, where the code for MET is located AUG. The anticodon UAC of the tRNA matches the start codon on mRNA AUG. The second codon in mRNA, which is GUU, matches the anticodon of tRNA carrying the amino acid valine, the CAA. The second tRNA binds the second active site on the large unit. A catalytic site on the larger subunit binds two amino acids together using dehydration synthesis, forming a peptide bond between them, met and val. The first tRNA now detaches and goes off to find another met in the cytoplasm. The mRNA chain shifts over one codon, placing the second codon CAU over the second active site. A tRNA with an anticodon GUA matching the exposed codon CAU moves onto the ribosome. This tRNA carries histidine or His. A new peptide bond forms between val and His on the catalytic site. The tRNA that carried val will detach and find another val in the cytoplasm. The mRNA strand then will shift over to one more codon. The process continues until the ribosome finds a stop codon. The subunits detach from one another, the mRNA is released, and the polypeptide chain moves down the ER for further processing. The initial met is removed and the chain is folded into its final shape. And here is your completed protein. And finally, we have the protein. And now let's have a summary of what we have just discussed. We discussed about transcription and translation. And just a review, we said that in transcription, a process by which genetic information encoded in DNA is copied into messenger RNA. It occurs in the nucleus, and the process is DNA to produce mRNA. In translation, the process by which information encoded in mRNA is used to assemble a protein at a ribosome occurs on a ribosome, mRNA to proteins. On this slide, I have two links to be used for simulation. This will help you better understand the topic that we have just discussed. Thank you. And if you have some question, please don't forget to shoot me an email and let me know whatever topic that I need to clarify. Once again, good morning.